Thank you. So thank you to all the student committee. This has been, I had high expectations of this. My expectations have been surpassed. This is a superb morning. And to think that there is an oncology, hematology and palliative care committee this active in Brighton is really phenomenal. So you've already done a lot of the work in setting the scene for what the Cancer Fund is about. We used to do a lot of building with the chemo suite. We use the conference suite, a third of the Horizon Centre. We've put in money for that. And we still do all the basics for patients. We do the teas and coffees, we do the car parking, we provide medical acupuncture for hormonal symptoms. We still do all those things you expect a local cancer charity to do, but we are looking more at the research side. We've heard this morning just how important research is. And we have immense skills and local abilities in research throughout the medical school, both University of Brighton, University of Sussex and the Trust. And it's a real pleasure to see it all coming together. So I'm a clinical oncologist. I've worked in my time across the whole patch, Worthing, Brighton, Princess Royal, Eastbourne, Hastings. It's an area which has really benefited from the medical school. And now that we've got a cancer fund which is looking and working with the committee, with the medical school, with the national research, we're going to hear just 3 minutes each, basically, from a string of researchers who are all currently or very recently funded. So I think we move on to that. And thank you everyone for spending their Saturday morning looking at this and anyone watching it again. So over to the presenters, and we're going to start off with Dr. Andrea Pepper.
Okay, thanks, David. So the sharp-eyed amongst you will note that I'm not Andrea, I'm Chris. Andrea is very lucky that she isn't me. But my job really is just as the warm-up act for Andrea and Eleni, who are going to talk to you about two specific projects that we're doing, which have been funded or are being funded by the Sussex Cancer Fund. So, James, can you just give me the first slide, please? Great. So Andrea and I arrived at Brighton and Sussex Medical School in 2017, in that glorious halcyon time, pre-COVID. And this is a picture of our team when we could get together and have barbecues and drink the odd beer. Hopefully those days will return. I'm ever the optimist. But when we first arrived, it literally was Andrea and myself. But you can see we've grown quite significantly, and we have plans to expand a little more in the future. But some at least of this expansion is caused or been helped by the Sussex Cancer Fund. So if you could give me the next slide, James. So this is an instrument that the Sussex Cancer Fund co-funded or matched funded with the medical school. So I'm grateful to both Malcolm as the head of school and to David as chair of the Sussex Cancer Fund for having the foresight to help us to buy this instrument. This unassuming looking piece of kit actually costs £180,000. Ouch, I hear you say. So the Sussex Cancer Fund provided £90,000 and the medical school the other 90, that allowed us to buy this piece of equipment. And it's proven its worth every single day. We use it every single day of our working lives in the lab. The picture on the right-hand side, if you just ignore the ugly fella in the background, the three lovely ladies from Sussex Cancer Fund, Jo, Jean, and Julia, are there in the front with Andrew, of course. And I'm going to hand over to Andrea now, who's going to talk to you about at least one example of how we use this instrument every single day to try to understand better some of the biological processes that go on in tumour cells. So over to you, Andrea.
Hi everyone. Can I have the next slide, please? So our group are currently working on 3 hematological malignancies at the medical school: diffuse large B-cell lymphoma, acute myeloid leukemia, and chronic lymphocytic leukemia. As Chris has just said, the Cytoflex is used for all of these projects and absolutely essential to our research. What I'm going to present today is some data I've got on CLL, and so that you can see how important it is to us. Firstly, I'll just give you a rough overview of CLL. It's the most common leukemia in the Western world and is currently incurable. It's a build-up of malignant but very dysfunctional B lymphocytes in the peripheral blood, the lymph nodes, and in the bone marrow. The tumour migrates between the 3 compartments. It's easiest to treat when it's in the peripheral blood, but it proliferates and is far more resistant to drugs when it's in the lymph nodes. It needs absolutely to migrate to the lymph nodes in order to survive, and therefore we need to try and block that migration. And next slide, please. So what triggers a CLL cell to migrate? Well, CLL cells have receptors on their surface that when that receptor is engaged, it can trigger them to both proliferate and to migrate. The B-cell receptor is one such receptor, and inhibitors of the B-cell receptor have absolutely revolutionized treatment of CLL. But unfortunately, some patients are refractory, some develop resistance, and it's not curative. The patients can never come off the drug. Therefore, CLL cells must be activated to migrate through another pathway as well. And our theory in our team is that pathway could well be the TLR9 or toll-like receptor 9 pathway. TLR9 is triggered by free DNA in the plasma. And our hypothesis is that actually it's the patient's own DNA that's triggering this. So basically what we think possibly happens is that the CLL cells themselves break down and die. They release their own DNA, and then that DNA actually triggers live CLL cells to proliferate and to migrate. And next slide, please. So I want to show you here some evidence to try and support that hypothesis of ours. Firstly, on the left-hand side, you can see a graph, which is the white blood cell count of patients with CLL against their plasmid DNA levels. And as you can see, the higher the white cell count of the patients, the larger the amount of the plasmid DNA they have in their blood. Interestingly, it's unmethylated DNA that actually triggers TLR9 signaling. So we measured unmethylated DNA in the plasma of patients with CLL and compared it with a normal healthy controls. And you can see from the middle graph here that patients with CLL, we found out, have up to 200-fold more unmethylated DNA in their plasma than the normal age-matched controls. The third piece of evidence is on the right graph, and it's a Kaplan-Meier curve of time to first treatment. Here we divided the patients into 2 groups, depending on whether they had low plasma DNA, which is shown by the blue line, or high plasma DNA, which is shown by the red line. On the x-axis, we've got the percentage that remained untreated, and on the y-axis, we've got the time to first treatment. And as you can see, the patients with the lower levels of plasma DNA, which is the blue line, survive much longer without treatment than those with high levels. Have the next slide, please. So next, I'd like to show you how we use the Cytoflex to identify CLL cells that will signal through TLR9 and then migrate. This is a patient with CLL cells that have been stained, patient with CLL whose cells have been stained with 7 colours. As Chris Mike mentioned earlier, our Cytoflex machine can actually stain up to 21 colours. Firstly, we use the scatter properties of the cells. So the CLLs or cells can actually scatter the lasers from this instrument in different patterns, and we can use that scatter pattern to actually identify the lymphocytes. So the first plot on the left-hand side labeled A is gating of the lymphocytes depending on their scatter properties. On graph number B, we have to make sure that we're only ever looking at single cells and not doublets, which are cells that are stuck together. So again, we use the scatter properties, the full scatter height against the full scatter area to gate on the single cells. Then we know when we get to C that we've got the single lymphocyte population. Lymphocytes are made up of B lymphocytes and T lymphocytes. And B and CLL is usually, not always, but usually a malignancy of B lymphocytes. Therefore, we use CD3, which is a T lymphocyte marker, to identify the CD3 negative B lymphocytes and gate on those. Then for D, we put in a viability dye to make sure that we're only gating on the viable cells. Dead cells will pick up the viability dye and fluoresce, and so we gate on the ones that are negative for that. And then finally, for graph E, we gate on the CLL cells themselves by using CD5 antibody and the CD19 antibody. The top right-hand corner shows that the CD5 positive, CD19 positive CLL cells are the main B lymphocyte population there. And you can see just to the left of those is a scattering of normal lymphocytes, which shows how dominant the malignant clone is. Once we've gated on the CLL cells, we can then look at their expression of TLR9 and also migration markers CD38 and CD49D. I've circled in red here the CLL cells that have got the highest expression of TLR9, but also the highest levels of CD38 and CD49D. These are the CLL cells that will migrate to the lymph nodes, and these are the CLL cells that we need to block. Have the next slide, please. Next, I'd like to show you how we can block migration. CLL cells from 7 patients being put into migration chambers, and the number of cells that migrated over a 4-hour period have been counted. The bar chart is a combination of 7 patients. In the royal blue bar chart on the left, you can see the maximum migration of the CLL cells in the absence of any drug. The turquoise bar shows that the migration is reduced by inhibiting the B-cell receptor with a drug called ibrutinib. Interestingly, when we inhibited the TLR9 on its own, the migration was even less than that inhibited by the ibrutinib, and that's shown by the red bar. Finally, when we combined both ibrutinib and TLR9 inhibition, we managed to reduce the migration the most. All of the counting for this experiment was also done on the Cytoflex machine. Here's the next slide, please. This data shows you how we've used the Cytoflex to show that TLR9 inhibition is an exciting new potential therapy for CLL. This work has now been published in the top international journal for hematology, Blood. In addition, it's also led to an MRC grant for 3 years so that we can carry on looking into this work. The questions that we're going to be asking from the TLR9, from the MRC grant, is whether we can use our simple migration model to predict which patients will respond to the current therapies out there and which patients need an extra drug as well, such as TLR9 blocking. And secondly, what we want to try and do with this grant is try and find a single drug that can block both the TLR9 signaling pathway and the B-cell receptor signaling pathway with one hit. And next slide, please. Finally, I want to introduce AML before handing over to Eleni, our PhD student. We have 2 projects on AML, both of which are using the Cytoflex really heavily. AML is a disease of poor survival rates, and the immature cells are anchored into the bone marrow where, again, they're protected from the drugs. Project One that we've got going is a really exciting project in collaboration with a drug development company in Uganda, looking at novel drugs to try and kill the stem cell compartment. And I'll now hand you over to Elaine, who will tell you all about her project, which is Project Two.
Hello, everyone. James, can I have the next slide, please? Thank you. So firstly, I'm going to introduce myself. So my name is Eleni. I'm currently a junior doctor training to be a hematologist. I also love research, in particular laboratory research, because I find it very creative. So the bench to bedside type of research. And thankfully the Sussex Cancer Fund has enabled me to spend 3 years in the lab to do my PhD on acute myeloid leukemia. My supervisors, you've already met them. It's Andrea Pepper, Tim Shavasu, and Chris Pepper. And basically my project looks at how the bone marrow environment affects the leukemia cells and how can we find new targets to improve leukemia treatments. Can I have the next slide, please? Thank you. So acute myeloid leukemia, as Andrea said, is the most common acute leukemia in adults with the lowest survival rate. It's relatively common. We have about 3,000 cases per year in the UK, and definitely its incidence is increasing since the 90s. The 5-year survival rate is quite poor at 20%, and for people over the age of 65, this can be as low as 5%. So there is a real need for better treatments. The big problem with AML is that even if the disease goes away with chemotherapy, it always comes, not always, but in most cases, it comes back, which is what we call disease relapse. Can I have the next slide, please? Thank you. So the theory behind my project is that the leukemia cells, these purple, pink dots on the figure, can move between the blood on the top in the vessels and the bone marrow on the bottom. In the bone marrow, the leukemia cells stick to other cells which protect them. So the chemotherapy goes into the patient blood, it kills the leukemia cells that are there, but it cannot reach and kill the leukemia cells that are stuck in the bone marrow. So over the years, the leukemia cells in the bone marrow grow and cause disease relapse. What I want to do is identify the mechanism by which the leukemia cell sticks to other cells in the bone marrow and develop new drugs to force them out of the bone marrow and then kill them with chemotherapy. Next slide, please. Thank you. So I have 4 aims in my project. During the first year, I'm going to focus on aim 1 and 2. Currently, I'm optimizing a 2D system that mimics the bone marrow environment. And to do this, I'm using different cell types to make it as realistic as I can. I'm going to test different drug combinations and find the best combination that detaches the leukemia cells from the bone marrow. And then later in my PhD, I'm going to optimize a 3D system now, mimicking the bone marrow, which is a lot more physiological and will actually mimic the circulation between the blood vessels and the bone marrow. Using this model, I'm going to compare the attached versus the detached leukemia cells and find novel targets from the cells that are persistently attached after all the drug treatments. Next slide, please. So these are just a few nice pictures from my experiments. You can see on the left, this is when on my 2D model, I plate the bone marrow cells. Then on day 2, I add the AML cells, and on day 3, I analyze the result. And on the right, you can see these bright dots. These are the leukemia cells, and you can see how beautifully they have stuck down on the bone marrow cells that are underneath. And a picture from further away, can I have the next slide, please? Is this one. So on the left, you can see the bone marrow cells that have grown in a plate. Then the picture in the middle is just leukemia cells on their own. And then on the picture on the right, you can see both of them together, and you can see how the leukemia cells grow on top of the bone marrow cells in patches. Next slide, please. Thank you very much. I mean, I just wanted to mention that the Sussex Cancer Fund funds me to do this work, so I wouldn't have been able to do it without them. But also my project relies on the machine, the Cytoflex, the equipment that Andrea and Chris kindly mentioned before. So I owe them a lot. Thank you very much for giving me this opportunity.
Hi, everyone. That's a picture of me. I'm not sure, can you see me?
Yes.
I thought I would just talk a little bit about what it is I do, and as a clinical hematologist, I have one foot, obviously, you know, in the hospital, on the wards where I see patients and help diagnose them and treat them for different types of blood cancer. But I also have a foot in the lab, and I work very closely with Chris and Andrea, who you've just heard from, and Elaine, obviously, who was an academic clinical fellow previously under me, and it's fantastic that we've managed to keep you, Elaine, in Brighton to carry on the important research that you're now doing as a PhD. So I have a long time interest really in acute myeloid leukemia, amongst other blood cancers. I'm also interested in CLL and collaborate with Andrew and Chris on that front. But my particular interest really is in acute myeloid leukemia. And the reason that this particular disease is one which I think, you know, deserves great focus on is largely because the treatments for AML haven't really improved, in all honesty, in about 40 years. Certainly in the time that I've been working as a consultant, the improvements we've made in survival of patients with this devastating diagnosis have been, you know, very small, and you know, and it lags behind a lot of the other blood cancers where there have been very significant developments in the last decade or two. So what we're particularly doing in my lab, which is based at the medical school and is shared really with Chris and Andrea, is to try and understand one particular type of AML, or I should say one particular type of mutation. And going back to what David said earlier about the mutations that underpin cancers, if we can understand the mutations, then we're going to be better placed to understand how best to target those mutations and improve survival in patients. And that's really the goal of the work we do. So the mutation, the gene that I'm particularly interested in is in a gene that's involved with regulation of the DNA, and more specifically, it is involved with something called methylation of the DNA. And I'm not going to go into too much detail about that, but I have here in front of me a model of DNA, and it's obviously hugely expanded in size. Your DNA is basically a whole list of letters and the mutation that happens in this one particular gene called DNMT3A is simply a spelling mistake in your genome. It's simply one misspelt word, so to speak, in the whole of your DNA. And if I were to expand this up to represent all the DNA in your cells, it would go around the Earth twice, I figured out this morning. And it's just one mutation in one particular base which causes the problem that can lead ultimately to acute myeloid leukemia. And what we've been endeavoring to do is to identify how this particular spelling mistake, if you like, how this mutation causes leukemia, because it seems to be a very early mutation, sometimes happening decades before the leukemia ever presents itself. How does that spelling mistake cause AML years later? And at the moment, we have glimpses of how that might happen, and it seems to involve the stem cells in the bone marrow, the cells which Eleni is looking at in her project, and the expansion of those stem cells over very many years to finally, declare themselves as this terrible cancer malignancy, which we know as acute myeloid leukemia. So we have, with funding from the Sussex Cancer Fund and indeed elsewhere, made some inroads here. And I think the first significant development we made was simply to set up a repository, a bank of samples donated by patients who we were treating in Brighton, of their leukemia cells, their bone marrow cells, which we have frozen down and store at the medical school in liquid nitrogen. And those that very kind donation really from patients has been a huge gift to us in terms of research to understand more about their disease, and more in particular about this particular mutation I mentioned, the one in this particular gene. We have found, two ways of targeting this particular mutation. And this dates back to work which again was funded by the Sussex Cancer Fund about 5 years ago, in which we identified, based on work that had been published elsewhere, two drugs that when used together were very successful in killing the mutated leukemia cells. And this is work we published, as I say, about 5 years ago. And we moved on to other research, but it was picked up by other collaborators elsewhere in Glasgow and actually in San Diego, in California. And through collaboration with those labs and using the patient samples, the bone marrow samples that had been frozen down here in Brighton, we managed to take this work on further to the next step, so to speak. And this work has recently been published in an important journal called Nature Communications, to show that using the two particular drugs that we first published on, that this really can make a difference in targeting AML cells that carry the particular mutation in the DNMT3A gene. And if we can get this into the clinic, then there's great promise that this might improve outcomes in patients in the future. So that's what we're doing at the moment. That's one sort of snippet really of some of the research we're carrying out. And there are various other strands of research which we're doing alongside that, as I say, to better understand leukemia and to improve our ability to target these specific mutations. I think I've probably taken up nearly all of my time. I'll just say one final thing, just really by way of introduction to Sally and Cresta, who I think are speaking next. And that is to say that as a clinician and a hematologist, and with my affiliation to the university. It's also been a privilege of mine to supervise other trainees coming through, both in haematology, as with Eleni, and also in oncology with Sally and Cressida. And I've been involved with oncology colleagues of mine in supervising their projects, and I'm looking forward to hearing what they're both going to say, because they have also done some fantastic work made possible by the Sussex Cancer Fund, and we are, hugely indebted to the generosity of the fund, and indeed the patients and relatives who have donated to the fund to allow this research to happen here in Brighton and across Sussex, where it's so enormously needed. So many thanks, and that's all I have to say for now, but very happy to answer any questions later. Thanks very much.
So my name's Cressida Lorimer. I'm one of the oncology registrars, and I train at Brighton and at the Royal Marsden. And I recently completed an MD that was funded, my salary was funded by the Sussex Cancer Fund. And they also funded part of my studies as well that I did as part of my MD. So I just wanted to just do a little quick chat about the work that I did as part of that. Next slide, please. So my whole MD was focusing on older patients with glioblastoma. And glioblastoma is the commonest primary malignant brain tumor, but still relatively rare in the grand scheme of things. But average age of diagnosis is about 64, and it's tending to get diagnosed more and more commonly in older patients. So the incidence is rising, and it peaks in about your 70th and 80th decade. And historically, as with a lot of cancer trials, older patients were excluded from the trials. Actually, more recently in the neuro-oncology field, there have been some randomized control trials looking at treatment options for older patients with glioblastoma. So there was some work that came out of Norway and Sweden and Canada that looked at patients over the age of 65 and different regimes in terms of surgery, radiotherapy, and chemotherapy. And so there are now treatment paradigms that are in place to give options for how to treat older patients with glioblastoma. Just to clarify, older is obviously quite a subjective term, and depending on what age you are, it may well depend on how old you feel old is. Neuro-oncology patients, because of the nature of the disease, so because of the tumor being within the brain itself, tend to age more rapidly than other cancer patients as a kind of general rule. So in a lot of breast cancer or gynecological cancer studies that are looking at older patients with cancer, often their cutoffs are more like sort of 75-ish or above. But actually in neuro-oncology, because of the insult that occurs to the brain through the tumor being there, the age that tends to be used to think about older patients tends to be around 65. So my project was looking at, although we have these ways of treating patients, so we have evidence to suggest that both chemotherapy alone, radiotherapy alone, and the combination of chemo and radiotherapy together have evidence behind them to help these patients in terms of their symptom control. It was looking at how we assess who's suitable for treatment. Unfortunately, glioblastoma is a pretty devastating disease. So even in younger patients, the average prognosis is only about 18 months. And although there is a tail of patients who have long-term survival, that's only about 5% or less of the whole group. And as you can imagine, as with a lot of other diseases, with older patients, the prognosis is poorer. And there was a big study that looked at data throughout the whole of England, which showed that actually in an all-comers group for patients over the age of 70 with glioblastoma, their prognosis was only about 3 to 6 months. And so whenever we're thinking about treatments for these patients, we have to balance their quality of life and the impact we're going to make on their quality of life with regular hospital visits, daily radiotherapy, side effects of chemotherapy, compared to the hopeful improvement in their survival that we may give them. But it's very much thinking about a palliative approach and how to recognize those patients that may benefit from treatment compared to those patients who you're just going to give horrible side effects to for their remaining months of life. Next slide, please. So I took a kind of three-pronged approach. So the beginning of my study, we looked at a retrospective review, so involving patients who had been treated for glioblastoma over the previous 5 years, and that was in Brighton at the Royal Marsden Hospital in Sutton and at the Beatson Cancer Institute in Glasgow. And we looked at a number of patients and tried to see whether there was anything we could tease out from their medical history and the symptoms they presented with in order to see if any of those were prognostic for survival. So no matter what treatment those patients have been given, were there any independent factors that could assess whether they could predict for how well people could do. And part of that also included looking at their tumour specimens. So from their original surgery that they'd had, their specimens were archived, and we got approval for these to be reanalyzed as in a lab at UCL in order to see if there were any particular mutations or anything we could see within this population that might also predict for survival. And that was part funded by the Sussex Cancer Fund with matched funding from the Beatson Cancer Charity and the Institute of Cancer Research. And that showed, so the pathological analysis on first analysis didn't actually show anything particularly unusual, but we are now working with a team in Belfast at Queen's University in Belfast who are looking at doing much more detailed molecular profiling of this than we did initially at UCL, and that work is still ongoing. The retrospective review we did showed that there were certain clinical factors as to how patients presented that could independently predict for how well they did. And so we used the background of this data to then design a prospective study. So my idea was that in a lot of other oncology subtypes, so especially in breast cancer and gynecological cancer, there's a lot of interest in doing what we call geriatric assessments. So instead of the oncologists just kind of giving someone the once over and going on their gut feeling in terms of whether they feel a patient is well enough for treatment, then using a bit more of a more detailed assessment technique. A true geriatric assessment should involve a whole multidisciplinary team. And we had to balance that in terms of real life, what was available in neuro-oncology clinics at the moment. And so I designed a neuro-oncology focused geriatric assessment tool, well, a geriatric assessment screen using pre-validated tools. And we did this as a prospective feasibility study in 3 sites. So again, in Brighton, Glasgow, and the Marsden, to see whether it was possible within the time and funding constraints of a busy NHS outpatient neuro-oncology clinic to achieve a neuro-oncology assessment. And this ran for a year and was really well subscribed to. It was run by the clinical nurse specialists in each group who really kind of took it on. It was very much their project, and they recruited very well, which was very handy for me. And so we got some good results of that and showed it was definitely feasible to do a neurologically focused geriatric assessment. Although the study wasn't powered for it, there was a strong suggestion that if people had a baseline impairment in their MOCA score, so in their cognitive assessment score, so looking at how their brain was working, then that was an independent predictor for how well they did. So patients who were very impaired in terms of their cognition and their functioning, not in terms of physical functioning, but just in terms of their processing and memory, no matter how those patients were treated in terms of how aggressively or less aggressively they were treated, they tended to do more badly. And so that was written up and published, and so was the retrospective review. And then my next kind of stem of this was to look at whether we could help try and predict who was more likely to get worse toxicity from treatment. So as I said, a lot of these treatments are based around quality of life. And there are some factors you can use from a lab point of view to see who might be more sensitive to chemotherapy. So there's a genetic mutation you can look for in these patients' tumours to see whether they are more likely to respond to chemotherapy or not. But we don't have a similar way of trying to assess who might be more or less suitable for radiotherapy. And so what I was interested in is whether we could look at using pre-treatment scans, so the MRI scans that a lot of these patients have as part of their diagnosis, to see whether we can look at the normal brain on those scans. And the theory being that, say, if you have a patient who's got high blood pressure, has potentially had a mini stroke in the past, then giving them radiotherapy to the brain may make them more likely to have acute side effects, as there's a theory that some of the acute side effects from brain radiotherapy are to do with how good your normal brain is. And so what we've done is we've set up a study that's still running at the moment, and that's running at about 13 centres across the UK. And that's looking at doing one extra MRI scan before these patients start their radiotherapy. And then they fill in, so they do a cognitive assessment, a questionnaire at baseline and at 4 weeks and 8 weeks. And they also do a quality of life questionnaire as well at that stage. And we're trying to see whether there is a correlation between changes seen on their scan before treatment and an impairment in their cognition or their quality of life. And the idea being that it wouldn't, you wouldn't ever say to a patient, you can't have radiotherapy because of what your scan looks like, but it would guide you a bit more to say, if we're thinking about treatment, actually, we've looked at your scan and we can suggest that you are much more likely to feel pretty shocking with radiotherapy and maybe not get the benefit of it. And therefore, we might go down a different line in terms of treatment. So in terms of chemotherapy or best supportive care. So most of my projects have been around trying to enable clinicians and patients to have as much information as possible and as much patient-specific information. So for their specific scan or their specific genetics, to see for them what their best approach would be in terms of trying to balance side effects and treatment benefit. And so, as I said, that study is still running, and that was very kindly also funded by the Sussex Cancer Fund. And we've recruited 100 patients. We just need 20 more, and that's due to close later in this year. So that's my sort of research synopsis. So many thanks to the Sussex Cancer Fund, who provided my salary and also a lot of the startup funding for some of my studies. I wouldn't have been able to do it without them or without a lot of the patients within Sussex and their families who have been part of my studies.
So I'm the last speaker. I'll try not to take too long. My name's Sally Appleyard, and I am similar to Cressida. I'm an oncology trainee, and I also work between Brighton and the Marsden. So this Cancer Fund had also funded my research time, which was 3 years spending time doing full-time research, and are continuing to support some ongoing studies similar to Cressida. And I think that's one of the advantages of the Cancer Fund for us, that similar to Cressida's work, we've been able to use local funding to start projects and then be able to access more funding elsewhere, because it's always easier to get funding for something when you've done some work already or when you've already got some money committed to it. So that's a really important role of the Cancer Fund in sort of what's described as pump priming these projects. It's actually worked quite well that Cressida went before me, because it, I think, demonstrates the whole spectrum of research that we do in Brighton. So there's obviously a lot of really important lab-based research, and then Cressida's research sits more within the clinical setting, so in the hospital, and a lot of my research is done within patients' homes. So I don't sit with a white coat on pipetting chemicals. I sit in people's houses and have cups of tea, which suits me very well, and it's a very enjoyable few years and a real privilege to spend time with patients and families in their own environment and get to know them in a different way than in the clinic. So my work is in bladder cancer and focusing on quality of life after treatment for bladder cancer. So bladder cancer is a relatively rare cancer. There's about 10,000 cases in the UK each year. And around half of those are invasive bladder cancer that has potential to shorten people's lives. unfortunately, similar to some of the other cancers we discussed today, the prognosis is pretty poor. And if people have got cancer that's invasive into their bladder, then even with the best and most radical, so most aggressive treatment, then at 5 years, only about half of them will still be alive. As with the other things we've talked about, it's really important to make sure that we are thinking about how people live their lives after treatment, as well as the treatment that we can offer them, so that we can make sure that we're not subjecting people to treatment that actually doesn't do them any favours. So the work I've done is looking at people who have invasive but localised bladder cancer, so cancer that we hope we can cure, and we can do that either by removing their bladder, and then people usually then pass their urine through their wall of their tummy into a bag, so a stoma bag, or they could have radiotherapy, which is X-ray treatment, and they have to come to hospital every day for 4 to 6 weeks to have this treatment. They get to keep their bladder, so they then get to continue to pass urine normally. So there are these two options, and we know that the outcomes in terms of cancer are similar between those two options. But what we don't really have a good handle on, and what there's no data at the moment, is how people feel, how they adapt, and what the changes that they experience after treatment. And some of the challenges for us as doctors and the surgeons making decisions is that, you know, we're only really able to spend time with the patients when they're having their treatment. So we have a good handle on the kind of what it's like in the immediate aftermath of treatment. But, you know, if they're well and at home in the months and years following, we don't really have an insight into how life is for them and the different impacts of treatment. Can you move on to the next slide, please? So the part of my study that fell within my MD, within my research project, was an interview study. So this was where I went and sat in people's houses and had tea and biscuits. And I spent time talking to patients, talking to their carers, so usually their spouses, and then also time talking to doctors and surgeons and specialist nurses, and really discussing with them how, so with patients and carers, what their experience was of treatment, both immediately after treatment and in the months and years that followed, and also about how they'd approach deciding between the treatment options if they were given an option between surgery and radiotherapy. It was very interesting speaking with patients and then reflecting on their experiences of treatment and how life has changed. And then being able to spend time with their spouses, which I always did on a different day, so that it wasn't all muddled together, and hearing their perspective on it. And then the sort of third angle of the clinicians and how they approach, really mainly how they think about what treatments they suggest to people. You can move on to the next slide. There's just a couple of quotes from patients. So one of the overwhelming themes that arose from the research, so when I do the interviews, they're recorded and then transcribed. I'm about to be interrupted by a child, I think, sorry. Jacob, go away, please. I'm just in the middle of something. Off you go. The patients, and then we analysed the interviews by looking at the things that patients have said and thinking both about them superficially and also at a deeper level, to think about how that all links together between different patient groups and the things that are different and similar between the patients and carers. And I think one of the main things was about people really put up with an awful lot of change and an awful lot of disruption to what they can do, because their perspective is, well, otherwise I would be dead. So that's the option, isn't it? If you've got a cancer that's going to kill you, you'll put up with a lot. And what that means is that sometimes when people come to clinic to see us, we say, how are you getting on? They say, I'm fine, thank you, because in their head they're thinking, well, otherwise I wouldn't be here. And I think that sometimes means that we as doctors and nurses aren't able to help people with things that actually we probably could improve, because people don't necessarily volunteer them. And I think without sitting in the patient's shoes and literally sitting in their houses, it's sometimes hard to explore those things. And then just move on to my last slide, please. So that was the study that was in my my research project, and then that has led on to a larger study which is part funded by the Cancer Fund, but much more funded by a drug company and a radiotherapy company. So the Cancer Fund funding allowed us to access that additional funding. And this study is looking at the same question really about how people live their lives after treatment for cancer, either with surgery or radiotherapy, but using questionnaires to quantify that a bit more, but very much looking at how people feel about their symptoms and what they're able to do. And we're running this in 35 centres across the UK. We've currently got nearly 250 patients recruited, and we're expecting to complete recruitment by the end of this year. And this data, because although it's really interesting to explore in depth people's ideas and feelings by talking to them, doctors and funders are interested in numbers primarily. And so having this questionnaire data for a large number of patients will allow us to explore better and present better arguments for changes in decision making and changes in the way we inform patients and support them. And that's everything I was going to say. I'm very sorry for the interruption. But anyway, and yeah, happy to hand back to James. Thank you.